My name is Gsponer. I'm associate professor here in the Department of Biochemistry and Molecular Biology and at the Michael Smith Laboratories. And I'm interested in recruiting a student who wants to study protein phase separation and phase separation mediated regulation of a virulence factor in Mycobacterium tuberculosis, which is the agent that causes tuberculosis. So my lab, in the most general terms, is interested in investigating the interactive behavior of proteins with large disordered regions. These are protein regions that lack a unique tertiary structure in isolation, but these regions, despite the fact that they have not a unique tertiary structure, they can still play very important roles, regulatory roles, and we're interested in how these so-called IDRs mediate intramolecular interactions and intermolecular interactions and thereby contribute to the regulation of the activity of proteins. And we're also interested in how they can mediate the assembly of higher order structures like amyloid structures, or more recently, dynamic protein condensates. In order to study this type of interactions, we use a variety of methods, mainly computational, but in the last couple of years, we've also started doing experiments, atomic force microscopy, and other types of experiments. So the system for which I'm looking a student for is this RV1747 virulence factor from MTB. So we got interested in this protein because this transmembrane protein has a cytoplasmic region that consists of two so-called FHA domains. These are the red domains here. And in between those two red domains, we have a very large intrinsically disordered region shown in blue. And so we were wondering how this intrinsically disordered region that is conserved across the family of these transporters contributes to the activity regulation. So how did we know that this disordered region would somehow contribute to the regulation of the activity? Well, it had been found previously that in this disordered linker, there are at least two phosphorylated threonines. And when one replaces these threonines with alanine, one gets the same phenotype as when removing the entire protein. And so what happens when one removes the entire protein from MTB? Well, one reduces virulence, meaning MTB would grow less in infected mice and macrophages. So we were interested in how this intracellular part of this transmembrane protein interacts with itself and how it contributes to activity regulation. What we knew already was that this protein is a transporter because it has a classical topology of an ABC transporter with a transmembrane domain and a nucleotide binding domain. So we first started studying this system from the cytoplasmic side, where we expressed constructs that contain both of these FHA domains plus the intervening linker. And with the help of nuclear magnetic resonance spectroscopy and molecular dynamic simulation, we studied what happens when one phosphorylates that linker between the two FHA domains. And to cut the long story short, we found that there is a very complex network of interaction of intra and intermolecular interactions. Most excitingly, what we found is that when one goes to very high concentration, so increases the concentration of this construct that contains both FHA domains and the linker, or when one phosphorylates it, in both cases, these systems starts forming droplets, condensates. So what are these condensates? Some of you might have heard of this. It has had quite a big impact in the last 10 years, protein phase separation. It has been shown that a variety of membraneless organelles in cells like P-bodies, but most prominently the nucleolus, are the result of protein phase separation, where proteins that are normally initially homogeneously distributed in solution coalesce and form these liquid-like droplets. as shown here in this video. These are droplets made of proteins that behave like water droplets. They can coalesce, they can grow, and they would sediment in vitro. So that was an intriguing finding, that this cytoplasmic part of this ABC transporter undergoes this liquid phase separation in vitro. So the question then is, what is this good for in the context of the entire protein? What is the result of the phase separation of the cytoplasmic part in the context of the entire transporter? Well, our hypothesis is that this phase separation or droplet formation leads to the clustering of multiple ABC transporters in the cell membrane, and that can increase, enhance activity or facilitate transport of the big substrates. Because we know also from the structure that this transporter is an exporter that exports molecule from inside the bacterium to the outside. And so we believe that the result of phase separation of the cytoplasmic part that enhances that export of substrates. But unfortunately, we do not yet know what the substrates are. To provide the first proof that indeed such clustering could happen, we use super resolution microscopy. And with the help of super resolution microscopy, I show you here the result. We could indeed show that this transmembrane protein, this ABC transporter, occurs in the membrane in the form of clusters where we have multiple molecules close in space together. So these were the first results that we generated and that we have published in a recent PNAS paper. We then just recently got a CHR funding to further study this system, and there are a variety of questions that we're interested in. We're obviously interested in understanding better how this phase separation takes place. What are the sequence elements here in the disordered linker that are necessary for this phase separation? How can we change them, manipulate phase separation? We want to get a better picture of the overall structure of this ABC transporter, not only of the cytoplasmic part. We want to figure out what are the actual substrates and how does phase separation affect substrate transport. And ultimately, the most important question is, how does this phase separation of the cytoplasmic part play out in the bacterium? How does it help the bacterium to be more virulent? How does it affect this phase separation, the activity of the transporter in the bacterium? And how can we ultimately change phase separation and change the activity of this protein? And do we then see an impact on MTB growth when we infect mice? So it will be a multidisciplinary project where a lot of people here at UBC, not only my group, will be involved, lots of techniques, biophysical techniques to study phase separation, to study the structure, to express the protein purified, and maybe with cryo-EM determine the structure, metabolomics approaches to identify the substrates, mutagenesis experiments to see how phase separation and the clustering is affected via mutations, and obviously quite a bit of microscopy to see within cells how this phase separation plays out for the cytoplasmic part alone or in the context of the entire protein. So if you're interested in working on this very exciting project that is focused on a very hot topic, protein phase separation, please don't hesitate to contact me.